in particular impaired formation of nets, neutrophil extracellular traps.
Good afternoon. I'd like to thank the organizers for this opportunity to present my work. I work in Professor Contino's lab at the University of São Paulo and start my postdoctoral fellow in this new topic of immunology. There's NETs, the neutrophil extracellular traps, and I think we all agree about the participation of neutrophils in bactericidal mechanisms and defend the host against pathogens. And the neutrophils, how can I point the mouse? Yeah, okay, that's right. But I think everybody agree about the importance of the oxygen species that neutrophils can release, about the elastase, myeloperoxidase, and the other peptides that neutrophil has in granules. And in the last few years, some groups of research start to think about another strategy of the neutrophils to kill bacteria and fungi. They call it NETs, neutrophil extracellular traps, that consists in the amount of DNA and nucleic material together with some bactericidal peptides. And they start to Thank you. Sorry, I think I was not nervous enough. So, but now, in the last few years, they show that some of pathways and some of mechanisms are important to net generations, to NETosis. Most of FOX, NAPD oxidase, and generation of reactive oxygen species, and the pathways that involve neutrophil elastase, myeloperoxidase, PED4, and histone citrullinação. And P38 was also appointed as an important pathway to NETosis and net generation. So since neutrophil extracellular traps is an important strategy, and in some cases this even the resolution of infections, we decided to analyze net generation in patients presenting severe fungal infections. We select 5 patients with chronic mucocutaneous candidiasis or severe chronic candidiasis, and 1 patient with APSED and with a mutation in R1 gene. From these 5 patients, we have 2 with mutations identified, 1 mutation in STAT1 and 1 mutation in CAR9, and we have 3 patients with no mutations identified. We start our trial through the analysis of DHR, because once the reactive oxygen species are important to NETosis, if those patients don't have a normal release of reactive oxygen species, it will be excluded of our experiments. So all patients have DHR, normal myeloperoxidase activity, but 3 patients, patient 1 with STAT1 mutations, patient 4 and patient 5, had no net formation when we incubate those cells, neutrophils, with Candida albicans. So to compare, we have the control group with non-stimulated neutrophils, stimulated with PMA, and stimulated with Candida albicans for 3 hours. The patient 1 with STAT1 mutation, neutrophils from this patient was able to produce nets when treated with PMA, but not when treated with Candida albicans. Patient 4 has the same a pattern of response. Patient 5 was not able to release the nets, but as we can see in this picture, we don't have lobulated nucleus of those neutrophils. And no response either against Candida albicans. This is the APSED patient that responds very well with PMA treatment, but not so well with Candida albicans. This is just a qualitative experiment, and we proceed in another experiment to analyze the release of nets in a quantitative way. So we use a EO-row. It's a DNA staining that do not pass across the plasmatic membrane. So they will mark only the DNA that is released. So we can quantify the nets. When we treated with PMA, we saw a failure in net release in these three patients, but not in the APC patient. But when we stimulated those cells with Candida albicans, we saw failure in release nets in all patients that, as we saw in the fluorescence microscopy. To complement our results, we test those cells to analyze the nets-dependent killing. Incubating neutrophils, just incubating the neutrophils with Candida albicans for 4 hours, these blank bars. So to analyze the total Candida albicans killing, depending on phagocytosis or not. And as we saw, those patients have a failure in Candida albicans killing. When we pre-treated those cells with cytochalasin D that impair the phagocytosis, we could see only the net-dependent killing. So as we see here, all patients have a failure in net-dependent killing. So the results suggest until now that STAT1 integrity is important for net generation. R1 is important for neutrophils' response against Candida. R1 integrity is important for net generation too. In summary, failure in net generation may be an important mechanism leading to susceptibility to Candida albicans. This is just a beginning of a research, and we pretend to identify not only the pathways that are important for net generation, but also new PID involving neutrophils. So I'd like to thank Professor Condino and all the groups that are involved in this work. Thank you.
I'll ask you one question. What was the purity of the neutrophils in your
My culture?
In your culture, yes. And also, did you do it in others? What serum was present in the culture with Candida and neutrophils? Was it autologous serum? Was allogeneic serum? Was FCS? Whatever.
The first question, the purity is over the 96% of neutrophils. And the second question.
Serum present in the culture. What source of serum? If there was any serum?
No serum. Okay. No serum.
Right. No other questions?
Thank you.